Hello and welcome to the course Basics of Experimental Design for Cognitive Psychology. I am Ark Verma from the Department of Cognitive Science at IIT Kanpur. We are in week 8 of the course and we are discussing event related potentials. Now, in most cases, ERPs actually originate as postsynaptic potentials which occur when neurotransmitters bind to receptors, changing the flow of ions across the cell membrane. We are basically in this lecture going to talk a little bit about where do these ERPs arise from, what is the source of this electrical activity that we are seeking to measure, and then we are seeking to make it contingent to stimulus processing within the brain. So the idea is that these ERPs actually are postsynaptic potentials which occur when the neurotransmitters are binding to receptors and they change the flow of ions across the cell membrane. The ERPs recorded at the scalps are not really produced typically by action potentials except in case of auditory responses that occur within a few milliseconds of the onset. When PSPs or postsynaptic potentials occur at the same time in large numbers of similarly oriented neurons, they summate, they are together and are basically conducted at nearly the speed of light throughout the brain. So, they traverse throughout the brain, the meninges, the skull and the scalp. Thus, ERPs are actually able to provide a direct, instantaneous, millisecond resolution measure of the neurotransmitter mediated neural activity. So, that is basically what we are measuring as ERPs. Now, when a postsynaptic potential occurs within a single neuron, it creates a tiny electrical dipole and oriented flow of current, and these measurable ERPs can be recorded at the scalp only when the dipoles from many thousands of similarly oriented neurons are put together or they are summed together. Now, if the orientations of the neurons in the given region are not similar to each other, then the dipoles will all cancel out and will be impossible to detect at a distant electrode. But if they are oriented to the same direction, so the main neurons that have this property are pyramidal cells of the cerebral cortex, which are the primary input and output cells of the cortex. These cells are all oriented in a perpendicular direction to the cortical surface, and their dipoles therefore they add together and instead of cancelling out. So, that is the activity that we can catch at the electrodes placed on the scalp. As a result, scalp recorded ERPs almost always reflect the neurotransmission that occurs in the cortical pyramidal cells. And basically what happens is that in non-laminar structures such as the basal ganglia etc., they do not typically generate ERPs that can be recorded from the scalp, and interneurons within the cortex are thought to generate almost little to no scalp ERP activity. So, only the fraction of brain activity actually leads to detectable brain activity in the scalp or detectable ERP activity in the scalp. Now, ERP components, just to sort of understand them, can be either positive or negative at a given electrode site. The polarity depends on the combination of factors. Say for example, it is usually impossible to draw conclusions from the polarity of an ERP component. So, you cannot really make a lot of sense of whether a particular component is negative, such as the N400 component, which comes over in reaction to sentences like he spread his bed with warm socks. So, when there is a semantic anomaly, this negative N400 component comes in. It is basically a component that peaks at 400 milliseconds in the negative direction. Similarly, we were talking about the P300 component in the last lecture, which is a positive component, peaks at around 300 milliseconds, and it basically is in response to the unpredictability of the stimulus. So, that P300 is positive and that N400 is negative. The positive and negative part of the component does not provide us much information. That is not typically what plays into the inference or interpretation of these components. Now, when the dipoles from many individual neurons sum together, they can be represented quite accurately within a single equivalent current dipole, which becomes the vector sum of all of these individual dipoles. If they are all summing together, they are oriented in the same direction. The voltage recorded on the surface of the scalp will be positive on one side of the dipole and will be negative on the other side, with a single line of zero voltage separating the positive and negative sides. Now, this voltage field spreads out through the conducive medium of the brain and the high resistance of the skull and the low resistance of the overlying scalp, leading to further blurring. So, it sort of blurs out over the scalp. Thus, the voltage for a single dipole will be fairly broadly distributed over the surface of the scalp, especially for ERPs that are generated in relatively deeper cortical structure. So, that is why the signal is relatively weak and it needs to be amplified. Okay, so we'll take one more example, an example of impaired cognition in schizophrenia that will basically tell us a little bit about how ERP components are used to isolate specific cognitive processes in the brain. Okay, so this experiment aims to ask why behavioral reaction times are typically slowed down in schizophrenia patients when they are asked to perform simple sensory motor tasks. Now, in these patients, RTs, could be slowed down because of an impairment in perceptual processes, or an impairment in decision making, or an impairment in the response selection process. So, we basically are interested using ERP to know what is it that is causing the slower reaction time responses, and we are basically studying it through the ERP methodology. Now, ERPs are ideally suited for answering this question because they provide a direct means of measuring the timing of the processes that occur between the stimulus presentation and the final response. Now, as per previous research, experimenters hypothesized that the slowing of the reaction times in schizophrenia in simple tasks simply does not result from the slowed perception or decision making, but it results from an impairment in the process of determining which response is the appropriate one, and once the stimulus has been perceived and categorized. So, it seems that this slowness of response is basically due to the time that is taken by the participants, by the schizophrenic participants, in deciding the appropriate response, in the response selection process. Alright, so this is the hypothesis that they went in with. To test this hypothesis, the experimenters recorded from 20 individuals with schizophrenia and 20 healthy control subjects in a modified oddball kind of task. In each 5-minute block of trials, the experimenters presented a sequence of letters and digits at fixation. Each stimulus was presented for a duration of 200 milliseconds, with a stimulus appearing every 1300 to 1500 milliseconds. Here, subjects were asked to make a button press response for each stimulus that they see, pressing with one hand for letters and with the other hand for digits. Now, one of these two categories was rare. Remember the oddball paradigm? So, one category is rare. So, one of these two categories was rare and the other was frequent. So, one came around 20% of the time and the other came around 80% of the time in any given block of trials. Both the category probabilities and the assignment of hand to categories were counterbalanced across trials. So, sometimes the letters will come 20% and the digits will come 80%, sometimes the digits will come 20%, letters will come 80%, sometimes you have to respond with right hand to the digit, sometimes you have to respond with left hand to the digit. Alright. So, these things were counterbalanced across the experimental block. Now, such a design allowed the experimenters to isolate the specific ERP components by means of difference waves, wherein ERP waveforms elicited by one trial type, let us say digits, was subtracted from the ERP waveform elicited by the other trial type, let us say letters, much like the difference images that we do in fMRI, etc. Now, difference waves are extremely useful in ERP research because they isolate neural processes that are differentially active for the two trial types. It basically tells us how the brain is separately processing the digits and how the brain is separately processing the letters. It eliminates the many concurrently active brain processes that do not differ between these two trial types. Okay. Now, this is important because the different ERP components are, ordinarily they are all mixed together, and it makes it difficult to determine exactly which component, which psychological or neural process differs across these different conditions or groups. So, basically what happens is the difference waves can pull out a subset of components, making it possible to draw more specific conclusions. This is where you can see this is the rare waves. You can see the rare P3 is obviously higher as opposed to the frequent P3, and you basically compute a difference wave. So, this is the rare minus frequent difference wave for P3, and this is what you can see. This is typically what is happening in the control subjects with the solid line, and this is slightly subdued or attenuated in the patients. So this is the rare P3, this is the frequent P3. You can see the rare minus difference frequent, rare minus frequent difference wave, and you can see that the waves or the effects are different for control subjects compared to the patients. So in the current study, the rare minus frequent difference waves were constructed to isolate the P3 wave, which is the the one that we're looking for in an oddball paradigm, which basically tells us about the time course of the stimulus categorization. Okay, that is the time it took the process of determining whether the stimulus falls into the rare or frequent category, alphabets or letters category, sorry, letters or digits category. Another set of difference waves was constructed to isolate the lateralized readiness potential. So this is another component that we are finding or looking for here, which reflects the time course of response selection after stimulus has been categorized, after you have decided that this is a letter or this is a digit. Okay. Now this LRP is also isolated by subtracting the voltages over the ipsilateral hemisphere on the same hand from the voltages from the contralateral hemisphere, the other hand. The experimenters found here that the RTs were slowed by approximately 60 milliseconds in patients as compared to control subjects, and the question was whether this this slowing reflected a slowing of perception and categorization, or whether it reflected the slowing of post categorization response selection process. Now the figure 1.4 basically shows this one. The figure 1.4 basically shows us that there is a slight difference here. Okay, the ERP elicited by the rare category, the ERP is elicited by the frequent category, and the rare minus frequent categories. We saw those three, and these are the grand average waveforms, which basically are first computed across trials for each subject at each electrode site, and then these waveforms at each electrode site are averaged across subjects. So that is why they are called grand average waveforms. These grand average waveforms basically make it easier to look at the data. So this is across trials for a particular subject and then averaged across subjects. So this is what is called the grand averaged waveforms. Now, as with previous studies, the voltage during the period of P3 wave, approximately that happens between 300 to 800 milliseconds, was reduced in the schizophrenia group, as we just saw, group relative to the control group. However, the voltage during this period is the sum of many different components, and it is not just that of the P3 wave. The rare minus frequent wave difference wave allows us to better isolate the P3 wave and to focus on brain activity that reflects the classification of the stimulus as belonging to the different category or the rare category or the frequent category. Now here you can see the patients exhibited no reduction in the amplitude of the P3 wave in the difference waves. You can see here again the amplitude is pretty much similar for the patients as well as the control subjects for the P3 wave in the rare minus frequent difference wave condition. Okay. The most important finding here was that the timing of the P3 was virtually identical in both patients and controls, which indicates that the patients were being able to perceive and categorize these stimuli just as fast as the control subjects, even though we see later that their reaction times are slower by 60 milliseconds. This slowing basically then it tells us that the slowing is not because of perception and categorization differences, but rather in response selection differences. So this implies that the slowing of the RTs basically reflects an impairment in the processes that follow post-stimulus categorization. Indeed, the LRP, which is an index of response preparation, was delayed by 75 milliseconds in the onset time, and it was much smaller. It was diminished by 50% in the amplitude for patients compared to control. So that's a different analysis. Also, the degree of amplitude reduction across patients was found to be significantly correlated with the degree of RT slowing. So here now you can see how we can use reaction time, how we can use the ERP methodologies to understand procedurally or the mechanics of how the schizophrenic patients and why the schizophrenic patients are slower in their behavioral responses when they are going through this oddball paradigm. So for a relatively simple perceptual task, the slowed RTs exhibited by the schizophrenia patients actually appear to result from primarily from a slowing of response selection as evidenced by the later and smaller LRP rather than the slowing of perception or categorization. This is a very good example of how we can use the ERP methodology to understand the mechanics of particular processes. Alright, so I will stop here and I will continue this discussion on ERPs, basically looking at the various steps of ERP experiments in the next lecture. Thank you.